For the last 50 years, researchers have been searching for a way to predict the 3D structure of proteins from the one-dimensional sequence of their amino acids. From the central dogma, we know that DNA is transcribed into RNA and then translated into an amino acid sequence, which contains all the necessary information to reliably fold into a three-dimensional protein. Researchers have successfully determined protein structure experimentally, using methods such as X-ray crystallography, nuclear magnetic resonance spectroscopy, and cryo-electron microscopy. But these methods are time-consuming and expensive. The Alphafold artificial intelligence system, developed by Google DeepMind, is the first non-experimental method that can rapidly accomplish this with accuracy comparable to experiment. But how does Alphafold 2, the 2021 iteration of Alphafold, speed up the process of obtaining useful 3D protein structure models? Here is a simplified diagram of Alphafold 2's architecture. We'll break it down into 3 sections and start here. When an input sequence of residues or amino acids is entered, Alphafold compares it to several databases of protein sequences to extract similar sequences from various organisms and tissues to generate a multiple sequence alignment, or MSA, and an initial MSA representation. It also pairs the input sequence and searches databases for templates of similar sequence proteins with experimentally determined structures. This is then used to create an initial pair representation of the input sequence, representing the relationship between every pair of residues within the target protein. From here, we can move on to section 2, the evoformer. The evoformer is a neural network unique to Alphafold. It consists of 2 towers that can communicate information to each other. For the MSA representation, the neural network prioritizes looking for row-wise relationships between residue pairs in the input sequence, before considering column-wise information that evaluates the importance of each residue in context of the other sequences. The pair representation tower evaluates the relationships between every 2 residues, which can be thought of as nodes, to refine the proximities or edges between the 2. It achieves this by triangulating the relationship of each node in a pair relative to a third node. The goal of this process is to help the network satisfy the triangle inequality theorem, where the sum of 2 edges on a triangle must be equal to or greater than the third. So how and where do the 2 towers communicate? Before the pair representation calculates the triangulations on each edge, it considers the updated MSA residue relationships and updates each edge accordingly. The results of the pair representation data is then used in the MSA's row-wise weighting of pair relationships in the input sequence prior to its next round of row and column-wise evaluations. These individual and cross-communicated calculations happened 48 times in the evoformer before creating the refined models of the initial MSA and pair representations. The final section involves another neural network called the structure module. It takes the refined models and performs rotations and translations on each amino acid, revealing an initial guess of the 3D protein structure. It also applies physical and chemical constraints dictated by atomic bonds, angles, and torsional angles. The refined models, as well as the output of the structure module, is iterated back through the evoformer